the other ones that are available. Just a reminder, the discussion, I've asked you to tell me in general what your discussion topic is, but it's not that I want you to discuss a topic in general. I want you to pick a very specific issue, question, problem, paper, something very specific to talk about within that topic. So you should, it's only 15-20 minutes, so you shouldn't spend a lot of time on introductory stuff, but get to the meat of the paper, problem, question, whatever, and make sure that there's some contribution that each of the team members can make. If for some reason one of the team members is not going to do a lot of talking, you can tell me how they contributed to the presentation or whatever. So, any questions about that? We had a number of people taking comp exams last week. Congratulations, everybody who got past that. I'm sorry to give a quiz on the second class, but since we're only meeting once a week, we don't have many opportunities. But I decided, because there were a lot of people missing, that I'm going to try to give all the answers to the quiz questions in the next hour or so. Last week, so if you don't get it right, it's not because you haven't heard it last week and this week. Okay, so what we're going to do, I don't think I can talk for more than an hour, and you can't listen for more than an hour, so I'm going to try to keep the lectures about an hour each, and then we'll have at least a half an hour for, we won't need a half an hour, but we'll have a half hour quiz and breath in between. I think probably going forward, we'll generally structure things that way. We'll do the discussions, quizzes, or whatever between the two lectures, just to give me a break. And I know Friday's tough. People have sports matches or visiting boyfriends or whatever, so let me know if you have to leave early on occasion, and I'll trust you to keep up with the material. So one thing I wanted to emphasize today, throughout the semester I was looking ahead at my slides, and I'm a very visual person. I don't like slides with a lot of words. I try to say the words and you can look at the picture, but I have a lot of slides that have a huge amount of content in them. So like every cytokine known to man or every chemokine known to man, I'm not showing it to you because you need to know those. It's just to give you a reference or an idea of the complexity. Everything we're learning is, you do have to know some specifics. You do need to know what kind of cell makes antibodies and what specific cells do, but when it comes to all the proteins in the complement system, all the chemokines, I'm not giving you that information because you need to know it. And because everything is take-home in terms of the exams, you have all the references you need. So I just am stressing that not to get worried by really intricate slides and try to focus on the main points. Okay. So the first thing I want to do is answer some excellent questions that were posed last time. So I think, so I actually can't remember who asked about boy in the bubble. So there actually was a boy in the bubble named David Vedder in Dallas. He lived from 71 to 1984. John Travolta then made it into a movie later on, but there's a good documentary on NPR about him, and a not so good movie by John Travolta about him. He suffered from severe combined immunodeficiency. He had no T or B cells. And so if you look at, we talked about hematopoiesis last week, starting with a common pluripotent stem cell that then becomes either a myeloid stem cell or a lymphoid progenitor cell. So then what David had was a deficiency of adenosine deaminase, so that's a required antibody to become B cells or T cells. So he had no adaptive immune system at all. That's just one of a number of immunodeficiencies. There are some that on the T cell side where people just lack T cells because they don't have a thymus or missing MHC molecules, and there are some people that just lack B cells because they lack essential enzymes on that side. And Kai asked me about chemical peels, which I knew nothing about, and now I do. So it's actually, I was just thinking of like scrubbing off skin or something, but they're actually cosmetic surgeons that do these, sorry?
Uses acid?
Yeah, acid and phenol, basically, to reshape people's skin. They do this for burn victims, so part of it is sort of therapeutic, but it's also done cosmetically. And it turns out it takes off all the skin down into the living skin layer, so getting down to the immune system we were talking about, the SALT, the skin-associated lymphoid tissue. And so there are a lot of infections caused by this process, as you suspected. So these are all the complications. You can just get dry or burning skin, which you can use lotion for. But if you happen to have herpes, if you get cold sores, then often you get a whole lot of cold sores after you undergo this. So people actually take antiviral therapy or antibiotics in order to be able to go through this process and not have problems. So, good question. This is why I bring the good questions, because then I get to learn stuff, not just you guys. Okay, so just a reminder, a lot of what we talked about last time was the somewhat artificial division of the immune system into the innate and the adaptive. The innate system is evolutionarily much older. It goes back long before mammals. The receptors are fixed in the genome. For the most part, we all have the same receptors. There's no genetic recombination, no change of DNA. All cells are identical, and they're conserved molecular patterns, so components of viruses and bacteria that are recognized. There's perfect differentiation of non-self for the most part, so we don't recognize anything of our own. And this is what happens in the fastest immune responses. The adaptive immune system has memory, so you get vaccinated, you maintain your immunity. There's a secondary immune response, so your immune response gets better over time. The receptors are encoded in gene segments that are rearranged, so this is a unique place in the body where there's actually editing of the genome. So the genome is changed when lymphocytes become mature. So each lymphocyte in your body doesn't have the same DNA as you had as an embryo, for example. There's a distribution of clonal cells, so cells are derived from a genetically identical parent and expand. And for the most part, the recognition is for a specific, very small molecular structure. Discrimination of non-self is not perfect, so we can have autoimmunity, and there's some differences in the effector cytokines. So these are the kind of things that one could put on quizzes. Okay. So how do pathogens get in? So a lot of it's you can imagine from your own life. Where do you get sick? You get a cold when the virus gets into your airway. You get pneumonia when bacteria gets into your lung. You get diarrhea or vomiting when a pathogen gets into your digestive tract. So basically anywhere where there is an opening or a mucosal surface that supports life, you can become infected with a pathogen. So in the airway, obviously, that is predominantly in the form of droplets. Not many viruses actually can survive being dried out, so usually you get a spray of sneeze from somebody else or contact with the surface, and things like flu and Neisseria can get in that way. So you can get meningococcus that way, or influenza. GI tract, obviously contaminated water or food, so you can have acute food poisoning this way or get a systemic infection. We all have had health class at some point or another, so the reproductive tract is also a good way to pass pathogens between individuals. And then, of course, if you've had mosquito bites. In this part of the world, you can get Lyme disease. In other parts of the world, you can get yellow fever. Sorry, Lyme disease is ticks, not mosquitoes. And then obviously, the most common effect is basically just breaking the skin surface. If you don't keep those areas clean, then infections get in easily through your skin. So the point really is that if we can maintain a barrier to the outside world, we don't need to induce any response. And the barrier is not just the epithelial cell, it's not just the tight connection akin to this castle wall, but there are also insulating layers of materials on different surfaces of the body that help prevent attachment of infection. So in the lung, there's tight epithelial junctions, there are cilia, there's a layer of fluid and a layer of mucus that traps things we inhale, and it's called a ciliated elevator. Essentially, there's a continual sweep of mucus from deep in the lung up into the bronchial tract and eventually into the GI tract, so you swallow this. Those of us who are polite swallow it, those of us who aren't spit it out. But essentially, the barrier is not just the living cells, but also whatever fluids are secreted outside. And those fluids have characteristics that make them antimicrobial. So, as I said, the tight junctions are important. So if you get any breakage of the tight junctions, that's a risk. But in addition to those tight junctions, there's also fluid flow. So there's a flow of air over the skin, there's a flow of fluid in the guts, there's movement of mucus in the lungs, sorry, and tears in the eyes. There are also chemical characteristics of those locations that make them antimicrobial. There's a lot of fatty acids. There's actually quite an acidic pH in the gut. So any pathogen that's going to survive to infect your lower GI tract has to make it through the pH of the stomach, which can be as low as 4 or 5. So several hundred to several thousand times more acidic than physiological pH. There's also enzymes in the stomach that are in the digestive tract that digest food, but they will also digest pathogens as well. The lungs have surfactants that have antimicrobial activity as well, and there are enzymes in the saliva and tears. Then there's a number of components of the innate immune system that are constitutively expressed at those surfaces that have antimicrobial activity, and we'll talk more about those as we move through. And in addition, this is especially important in the gut, we have a huge normal microbiome. So as I suggested before, last time there's about 2 pounds of microbes living on or in most of us, and the absence of those occupying the environment and providing their own antipathogen products can lead to serious infection, especially in the gut. People who go in the hospital and have to get antibiotics, sometimes their normal flora is cleared out, and that's an opportunity for pathogenic bacteria to grow. Okay, so again, the most frequent example that we experience often is a skin infection. So the different degrees of burn depends on how far into the skin one is burned by heat source, whether it's just in the epidermis, into the dermis, or through the entire skin layer. And obviously, the threat of infection increases as the burn goes deeper. So thinking about the last slide, there's a number of reasons that this could be infected. We lose the normal bacteria that are growing on the skin. We have access to living tissue as opposed to the surface of the skin, which is our dead cells. And there's loss of an effective blood supply. So there's low oxygen tension, which is good for anaerobes to grow. There's necrotic tissue, which is a good source of food for some pathogens. and it makes it harder to deliver immune cells to that tissue. So obviously, key in treatment of many burn patients is basically continuously removing dead skin. Okay, so in those situations where we're exposed to a pathogen in the lung, in the skin, by those breaks, an effective immune response keeps those from becoming systemic immune infections. So this is a bit artificial, but there's a stepwise process of a local infection involving adherence, gaining access to the living skin compartment, and then the kinds of inflammatory responses we talked about last time in terms of encountering macrophages in the skin, which have TLRs and can release local inflammatory cytokines, and then antigen-presenting cells, like dendritic cells, which can then present at antigen in the adaptive immune system, which goes on in the secondary lymphoid tissues. So in the secondary lymphoid tissue, then that antigen will encounter lymphocytes that have matured and can then be exposed to any pathogen-associated antigens, and then you can have an adaptive immune response involving antibodies or T cells that are specific for that pathogen. So it doesn't have to go through the whole cycle, but in order to control the thousands of local infections we get in our lifetime, this is the process that goes on. If an infection becomes systemic, that's when we're in real trouble. We'll talk about that in a minute. So we were talking about the lung and the importance of the mucus layer. An example of how important the mucus layer is to innate immunity comes in cystic fibrosis patients. So cystic fibrosis patients have mutations in a channel that transports chloride out of the epithelial cell into the extracellular space. And the reason that chloride is transported is that it makes an osmotic gradient. So that provides a way to move water out onto the lung surface. So that hydrates the layer where the cilia work under the mucus. So in the normal airway, there's a respiratory epithelium with its cilia and an aqueous layer on top of the cilia supporting the mucus, and then the airspace where particles and bacteria can be inhaled. In a normal individual, if bacteria get through that layer, they're exposed to the innate immune system, and there can be response by neutrophils and release of chemokines to recruit more cells of the innate immune system if necessary, and clearance of the infection. In the cystic fibrosis patient, there's very little aqueous layer, which results in the fact that the cilia cannot beat effectively in an aqueous layer, and the mucus layer builds up. So there's accumulation of pathogens in the lung, which provides an increased inflammatory signal and recruitment of more inflammatory cells. And that can lead to increased damage to the lung or just perpetual infection. So there's no defect in the immune system of cystic fibrosis patients, just this channel that transports water, but it results in a huge liability in terms of chronic lung infection.
Sorry, why does the aqueous layer help?
It's actually there's sort of a lot of hand waving about that, but basically the cilia beat effectively in fluid of a certain thickness. And right, so basically they move in that aqueous layer that's quite isotonic, and the mucus floats on top of that. When there's not enough aqueous layer, the cilia become tangled in the mucus, and they can't basically move along. That's the model anyway. Anybody else?
What are some of the possible treatments?
So, historically, there's been a lot of antimicrobial treatment. So when cystic fibrosis patients get a lung infection, pneumonia, then they're treated with antibiotics. They also have bronchodilators like asthmatics take to try to keep the airways as open as possible. There's been attempts, and I'll do a little more digging for next class. I haven't looked into it for a while. There's also been attempts at inhaling acetonic liquid mist to try to liquefy the mucus. There's been a lot of physical therapy, so a lot of pounding, basically to physically move that mucus and cough it out. There's also a lot of work on gene therapy, basically trying to be able to get cystic fibrosis patients to inhale a virus that infects their lung cells that then express an effective CFTR. And actually, we have, in my building at Pfizer, there is a rare disease unit, and they have found that there are actually some small molecule drugs that increase the function of the channel. So they're actually testing basic, just a pill that you would take that would improve the function of the CFTR. Because it's not that it's not expressed, it's just that it doesn't function very well. And I will bring myself up to speed by next class. Okay, so we've established that you want to keep a barrier to the outside. You have an immune system that becomes induced if they get through those physical barriers. You can have an induced response with an innate immune system. But obviously, when we get a skin infection, we know we have a skin infection. So why does infection cause disease? What do we experience from the infection? So there's direct tissue damage that the pathogens themselves cause, and then there are indirect mechanisms. Our own immune response damages tissues as well. So many bacteria release exotoxins. They release soluble proteins that are toxic to the cells around them. That helps them fight off the immune system and establish a place to live. So it's not really part of this course, but if you take your bacteriology, you'll learn all the fascinating toxins that bacteria can make. It includes tetanus toxins, so we're all immunized against tetanus because toxins released by Clostridium tetani would paralyze us. Botulism is another toxin released from bacteria. So they have very specific toxic mechanisms. Then there's endotoxins, so the coating of bacteria, that's the ligand of TLR4, which we talked about briefly last time and we'll talk about more today. That has direct toxic effects as well. And then there are lots of viruses that kill the cells that they infect. So the life cycle of a virus requires using cellular machinery, and some viruses kill the cells that they've infected. And then throughout the rest of the course, we'll be talking about immune responses, but obviously there's a lot of bystander effect to our immune responses. We talked about neutrophils undergoing oxidative burst, and they kill bystander cells. There are CD8 positive T cells we'll talk about later that directly kill virus-infected cells. And then immune complexes can cause a lot of the large connections of antigen and antibody can damage tissue as well. And when we get to autoimmune diseases, we'll talk more about that. Okay. So those are general concepts about how pathogens can get in and how they cause trouble in the tissues they infect. We're talking about innate immunity today. Really, the rest of the course will be about adaptive immunity. So today we'll just focus on how does the innate immune system, once the physical barriers have been defeated, how does the innate immune system know that we have been infected? And so, like we said in that big table, the pathogens have pathogen-associated molecular patterns, and those are recognized by pattern recognition molecules that are evolutionarily ancient in our innate immune system. So the components of pathogens that we can recognize are either their proteins, their lipids, they are nucleic acids in forms that we don't express, sugars, etc. So basically, any component of a pathogen can be recognized by pattern recognition molecules. The molecules that the innate immune system used to recognize those are in a number of classes. Some are cell-associated, like the toll-like receptors and the nod-like receptors, we'll talk about in a minute. Scavenger receptors and some other receptors we won't talk about until we get to NK cells. And then there are extracellular pattern recognition molecules that operate independently of cells, which include coplinates, lectins, and defensins that are discussed in chapter 2 of the book. Okay, so I used this example last time. The first toll receptor was discovered in '96 when it was knocked out of Drosophila, and they died by fungal infection when the receptor was knocked out. Even though the names are different, the signaling pathway that the toll receptors use in humans is essentially identical to that used in Drosophila, even though the proteins have different names. They have very similar sequences and functions. So the extracellular portion of the receptor binds to the pathogen-associated molecules. Then there's a number of associated signaling molecules intracellularly that eventually leads to cascade, resulting in a transcriptional response, which we'll talk about more in a minute. And that's essentially unchanged from flies to us. So while the Drosophila just had one toll, we have at least 10 TLRs, and they have a variety of different PAMPs that they recognize. And a lot of them recognize pathogen coats, like TLR1, TLR2, TLR4 are all coat proteins that are recognized, either of fungus or bacteria. Some of them also recognize some endogenous proteins that are released as well, like HSP70 heat shock proteins. TLR5 is specific to flagellum. It's a protein in the tails of flagellated bacteria. And then there are a number that are focused on nucleic acids that are unique to pathogens, like TLR3, 7, 8, and 9. So unmethylated CpG DNA is common to bacteria. Single-stranded and double-stranded RNA are common to certain viruses. Some of these can also be activated by small molecules that have been identified. So all these molecules listed here are synthetic compounds that have been identified that activate this receptor, and they use those as adjuvants, actually, basically to stimulate the immune response for vaccines.
Isn't Taxol like a anti-cancer compound?
Yep.
Or is it just a coincidence, or?
So, I don't know the basis. So Taxol is a natural product. It's purified from bark of some kind. Things in this chart are basically have just been put here because somebody wrote a paper showing that you could activate that particular TLR, that molecule. I don't know the basis of that. It's basically just that someone has shown that you could activate that receptor with that molecule. In the case of TLR7, drug companies were specifically looking for molecules that would activate TLR7. But a lot of the other ones are happenstance. And there are many more things that have been described as activators of TLR4, but it turns out that it's very hard to purify anything totally free of LPS. So there have been a lot of publications retracted once whatever it was that they used to stimulate TLR4 was purified such that all the LPS was removed, then it's no longer an activator of TLR4. But there are proteins other than LPS that do activate. So as I was saying up here, some of the pattern recognition molecules can also be recognized by DAMPs. So those are proteins that the body itself releases in the case of tissue damage, including those heat shock proteins. And there's always new toll-like receptors being identified. This is actually a paper from a postdoc in my lab, and with a collaboration with the group in Dublin, identified this protein TRIL by sequence homology that has an extracellular domain that's very similar to TLR4 without signaling complex. And went on to use siRNA to knock down the expression of that protein, which he called TRIL, and that had an effect on the inflammatory response to LPS. It's still not known how expression of TRIL helps the inflammatory response, but it's just a point that there are still, the first toll was identified in '96, and there's still more to be identified. Okay, so as that paper highlights, a number of the TLRs do not operate as standalone proteins. They have co-activators, co-receptors. For fully efficacious response to LPS, TLR4 requires requires CD14 as a co-receptor and MD2 as an accessory molecule. If you take MD2 or CD14 out of the picture, much less inflammatory cytokine is produced, and that helps maintain the specificity of the receptor because there are now 3 molecules that have to recognize that LPS. TLR4 is somewhat unique in that it activates 2 important signaling cascades. The details of these are not critical, except one, which is based on the adapter protein MyD88, leads to NF-kappa B and inflammatory cytokine synthesis. Many of the TLRs, or even most of the TLRs, can activate this signaling pathway. It also activates another set of signaling molecules using the adapter protein TRIF that leads to an antiviral response. And we'll touch on that at the end. But these are basically the 2 canonical signaling pathways that result from TLR signaling. And what varies between cells is which TLRs they express, because there's no cell that expresses all of them. So, just to make the story more complicated, not only do all the TLRs respond to different PRMs, but they're also expressed in different places. But this makes some sense. All the ones that recognize bacterial and fungal cell wall components are expressed on the outside of the cell, where they're going to encounter extracellular organisms. All the ones that recognize pathogen-associated nucleic acids that you wouldn't expect to be extracellular. You wouldn't see those until a bacteria was phagocytosed and digested in the cell, are expressed in the endosome. So the endolysosome is where phagocytosed particles are digested. So they're expressed right in the place you would expect there to be nucleic acids from inside pathogens. And then in the cytosol, so not associated with the membrane, are the other pattern recognition receptors, the RLRs and the NLRs. There's no real logic to how they're named. The RLRs are the RIG-like receptors because RIG was the first one of that class identified. The NLRs are the NOD-like receptors because NOD was the first one in that class. But those basically recognize components of pathogens that make it into the cytosol, so they're not either extracellular or within a phagolysosome.
Are TLRs always membrane bound?
TLRs are always membrane bound. Yeah, so that's the kind of distinction that you need to remember. I would say, not which TLR binds which ligand, but that you can just differentiate between TLRs and other receptors and whether they're membrane bound or not. And so, even though those are distinct systems, what you can realize is the specificity of the TLRs, the RLs, or the NLRs means that there's multiple pathogen recognition receptors for components of different pathogens. So the viruses, different components of the virus, whether it's DNA, RNA, or glycoprotein, are recognized by TLRs, RLRs, NLRs, and DNA sensors. And then as you go across, you can see which components of gram-negative bacteria, gram-positive bacteria, funguses, and parasites can be recognized by which. So it's just to point out that it's a highly redundant system, depending on what the pathogen is and where its components are exposed in the cell. Okay, so I'm just going to tell you comprehensively to finish the story about a couple of other cell-associated pattern recognition molecules. The specifics become less important. TLRs are most important. It's important to know that there are cytoplasmic pattern recognition receptors that sense components of pathogens. There's a whole bunch of different receptor classes on the surface of cells, especially macrophages, that are called scavenger receptors. They're of a number of different classes, but basically they have the capacity to bind things extracellularly and facilitate phagocytosis. They can bind sugars, they can bind LPS, they can bind almost anything. And these scavenger receptors are key for cells like macrophages that take up pathogens or dead cells or any other extracellular components. And again, these are not always expressed on the same cell in the same place. But just know as a class that there's a role for scavenger receptors in the immune system. And just on this slide, I'm just summarizing that there's a literature that some of the scavenger receptors, especially the class A scavenger receptors, have been shown to be important in responding to bacterial components. So if you make a mouse that's knocked out of scavenger receptor A or Marco, they're more susceptible to infection and they're less capable of taking up bacteria, especially in the lung. Okay, so again, it's the day of big lists. So we've talked about big classes of cell-associated pattern recognition molecules. Now, I'll quickly go through a couple classes of non-cell associated, of soluble pattern recognition molecules and the functions of those classes. And then we'll finish off the first lecture with a complement. So collectins are a whole bunch of similarly structured molecules, either in plasma or in the circulation, that bind to pathogen-associated sugars. So basically, they help to identify molecules that are foreign. You can see this in this table, all the different sorts of things that those collectins can do. They can agglutinate, in other words, they can glom the bacteria or their sugars together, which helps with phagocytosis. They can activate complement, which we'll talk about in a few minutes. They can have direct antimicrobial effect. They can regulate other cells of the immune system. They can help with clearance of cells. So this is basically, as a class, the collectins identify foreign sugars and make them easier for the immune system to see by one mechanism or another. Okay, so similarly, so the lectins are soluble proteins that help identify pathogen-associated sugars. Defensins are a whole class of molecules that help with the antiviral response. Some of them directly interact with viruses. Some of them are produced by cells that are infected with viruses to inform the immune system that a virus is present. Some of them recruit or regulate cells of the immune response to viruses. So Again, the specific functions of each of these is not important. But what's important to know is some are made by immune cells, like dendritic cells, monocytes, and macrophages. Some are made by the epithelial cells of the tissue themselves, and they regulate, they have specific functions. Some are constitutively present in mucosal surfaces, and some are induced in response to viral infection. But all of their effects are coordinated to limit viral spread. So this is just a cartoon of some of those molecules. So in general, they don't all do the same things, but the general functions of defensins are to inhibit the association of virus with the mucosal epithelium. So HBD2 and 3 are in that fluid in the lung or other mucosal surfaces, and they inhibit binding of the virus to epithelial cells. If a virus does bind to an epithelial cell, that epithelial cell is stimulated to make more of those defensins, and those defensins then have effects on leukocytes. So it downregulates the chemotaxis of lymphocytes, so they stick around. It activates APCs so that they're more capable of presenting antigens. It stimulates the release of chemokines and cytokines and activates neutrophils to make more defensins, basically. So it's a highly evolved class of stimulated and constitutive antiviral soluble proteins. So just understand the class as a whole. Any questions?
So that mechanism was humoral?
Humoral, it's soluble. Humoral generally applies to antibodies, the term humoral. So it's soluble, it's not cell associated, but humoral generally implies antibodies.
I'm just a little confused at the difference between defensins and collectins.
Okay?
Are defensins different because they can actually fight off the virus? Does collectins just kind of flag it?
So it's really just the targets. Defensins are predominantly focused on viruses, and they have effects on virally infected cells, immune cells, and the virus themselves. Collectins are specific binders of pathogen-associated proteins, so they don't have all those other effects when they're not associated with the pathogen. Collectins basically tag a pathogen for some immune activity, whether it's phagocytosis or complement activation. So defensins are a little bit more of a broad class. They target self and pathogens, and they have a variety of functions. Collectins basically have one function: to bind pathogen-associated sugars and activate the immune system. But in general, they're both soluble pathogen recognition molecules. Okay. So the last piece that I want to talk about before the break is complement. It's an incredibly, taken as a whole, it's an incredibly complicated part of the innate immune system, but it's the basics of what it accomplishes are fairly simple. So I'm hoping to try to digest it into a logical progression that you guys can understand. Again, knowing all the names is not important. What's important is to know is what can activate the complement system and what the end result of activation is. It's a cascade of enzymes that act on other enzymes, zymogens. So they're inactivated enzymes that become activated and then activate other enzymes in the pathway. So it has an incredible capacity for amplification. So a little bit of signal on the top can result in a lot of signal on the bottom. And it's highly regulated. We'll get to some of the regulatory proteins in the beginning. Like Collectins, it's a system for activating the immune system when a pathogen is present. So it does a lot of important things by binding to pathogens. It can increase recognition and optimization. So a pathogen that is coated with complement is easier to phagocytose and easier for a macrophage to identify. So there are receptors on immune cells for activated complement proteins. Again, as I said, by decorating the outside of the pathogen with complement, it's much easier for pathogens to be taken up. Components of the complement system are released as small diffusible molecules and lead to chemotaxis. So activation of complement leads to recruitment of inflammatory cells to the site of infection. And the downstream parts of the complement can actually form a pore on the surface of a pathogen or an infected cell to lyse that cell. So they can be directly cytolytic to pathogens. So complement can help aid both the innate and the adaptive immune system and both cell-mediated and antibody-mediated immunity, which I'll describe in a second. So the pathogen, the complement cascade is basically divided traditionally into three pathways called the classical, the lectin, and the target pathway. So these aren't separate systems. They basically describe the fact that three different pathways can activate the same enzyme cascade. So classically, the classical pathway activates the cascade by the combination of antigen and antibody. So you can think about a bacteria comes into the tissue. We have formed an immune response against this bacteria before, so there's an antibody around against that bacteria. The antibody deposits on the bacteria, and because of the antibody-antigen interaction, complement is activated. And the activation of complement helps recruit more immune cells. It helps deposit complement on that bacteria, which makes it easier to phagocytose, and it activates other receptors of the immune system. So, and feeding into the same complement activation is a lectin pathway. Basically, it's binding of lectins, or those collectins, to sugars on the pathogen surface. So similarly, that activates the complement cascade. And then there's this pathway called the alternate cascade, but it's essentially direct activation by some unknown mechanism of the complement cascade by the pathogen itself without the requirement of a lectin or an antibody complex. Simply the proximity of complement to the pathogen spontaneously starts the activation of complement. So the actual cascade is the same in all cases. It's just the stimulus for the activation of the cascade that's different. And the downstream results are all the same. Recruitment of inflammatory cells, opsonization, and direct killing.
What's opsonization?
So optimization is making it more visible to the immune system. So it basically makes it easier for receptors of the immune system to identify. So now the pathogen doesn't just have LPS on the surface for TLRs to see, but it also has deposited complement. And macrophages have a whole bunch of those pattern recognition receptors on the surface. It has receptors for complement, receptors for LPS, scavenger receptors. So the more interaction between ligand and receptor, the more likely it is for the pathogen to be taken up. So when you reduce all those different pathways to one cartoon, it looks like this. So they're not three separate pathways. The antigen, depicted here in yellow, can be recognized either by antibodies that bind the C1 complex, or it can be identified by the lectin pathway. So it can be bound by lectins or by antibodies to activate a C1 complex, which the C1 complex then cleaves C2, which cleaves C3. And so each cleavage, basically the piece of protein that's removed allows enzymatic activity, and each complement enzyme has specific activity for the next complement protein. So the classical and the lectin happen up here, and they lead to activation of C3. Once you're at C3, everything else that didn't involve lectin or antibody is the alternative pathway. And so a lot of proteins are not being shown here, but C3 is basically where you're committed to go down the rest of the complement cascade. C3a is a chemotactic protein, as is C5a, and then C5b through 9 form the membrane attack complex that lead to that direct swelling. So all those processes we talked about, identification and opsonization of the pathogen, recruitment of other inflammatory cells by release of C3a and C5a, and then direct killing, are all happening in the same cascade, although they're activated by different pathogen recognition molecules. Okay, so I'm just kind of hammering on the same point. Classical cascade is assembly of that C1 complex. So this C1 complex is not assembled until an antibody is bound to antigen, and then this becomes an active enzyme and cleaves C2. So this is classical. And then surprisingly, similarly looking is the lectin pathway. So when lectins are bound on pathogen-associated sugars, they activate the same pathway. And then again, the alternative pathway, which probably happens, it may be the most frequent activation, is simply some physical interaction between C3 and the pathogen surface directly activates C3. And there's, again, lots of proteins you don't need to know about in this process. So this table just summarizes all the things I've been talking about, the different functions. So basically, it's adding the different components into the classical lectin and alternative pathway, and tells you what actually the function of each of the downstream proteins are. C3a and C5a are chemokines, they recruit cells. C3b directly interacts with the pathogen and basically determines where the membrane attacks complex is going to happen. And the last piece is, you can think about, this is an incredibly powerful system. The complement proteins are in huge concentrations in the plasma. So there's a huge enzyme cascade poised to be triggered at any given time. And so you can imagine that it's pretty important to maintain the quiescent state of complement at all times. So there's a whole bunch of proteins that bind C4b, proteins that bind C3, and proteins that bind the lectins or the upstream processes to basically inhibit the activation of the complement system. So in order to activate the complement cascade, you have to overcome all those inhibitory signals that prevent the cascade from activating. And then this is all of those players in one table, if you'd like to look at it that way. But like I've been trying to repeat the important points, how you activate the complement cascade. In general, what types of proteins those are, those zymogens that are activated by upstream enzymes, and then all the downstream functions of complement. So this is just a summary of the first part. And basically, we've gone through the best way not to get infected is to keep pathogens out. This is done by maintaining your epithelial barrier and all the fluids on mucosal surfaces. Those mucosal surfaces contain some constitutively released antimicrobial substances like lysozyme, defensins, like maintaining a low pH. In the case that pathogens do get through into the tissue and an infection occurs, all of these components become exposed to the innate immune system. We have a long list of pattern recognition molecules associated predominantly with inflammatory cells to identify those molecules and either activate the adaptive immune system or directly deal with the pathogen itself. So, any questions on this part? Blank stares are good? Okay. It's 2:05. Take the quiz and go to the bathroom in whatever order you want. We'll start again at 2:40.